Sugar replacements, known generally as artificial sweeteners or non-nutritive sweeteners, have been in the news a lot. Rising concerns exist about safety and the effects of the sweeteners on many key features of health, including the microbiome. We need the help of talented scientists to sort through this complex web of information. Welcome to the Leading Voices in Food podcast. I'm Kelly Brownell, Professor of Public Policy at Duke University and Director Emeritus of the World Food Policy Center at Duke. Today's guest, Professor Eren Elinav, has done some of the seminal research in this area. He is an expert on systems immunology at the Weizmann Institute of Science in Israel. Dr. Elinav, thanks so much for being with us today. So back in 2014, you and your colleagues published a study in the prestigious journal Nature showing that the sweeteners could induce glucose tolerance, a highly concerning finding, and that they did so by changing the microbiome. So would you tell us a little bit more about this study and what subsequent researchers found?
In the study that we published in Nature back in 2014, we explored the consumption of several commonly used artificial sweeteners when administered to mice. And we wanted to ask a very simple but important question, which is, are those formulations of hyper-sweet compounds, which are so massively consumed by humans, in fact being responded to by our gut microbes? And we know from our other studies that gut microbes contain a very well-developed machinery that enables them to respond and to digest multiple very complex formulations of food. And so we asked ourselves whether these seemingly inert compounds could induce changes in the gut microbiome, which could impact the health of these mice. And indeed, when we tested this, and we mainly focused on saccharin, but also in several experiments, saw that this could also be true for other commonly used artificial sweeteners. What we saw was that the gut microbiome of mice being given saccharin at different concentrations, different strains of mice, and different obese or lean conditions were indeed capable of reacting to saccharin, changing their composition and behavior accordingly, and could, in some cases, with some mice that harbor different signatures of microbes, resulting in a very surprising and counterintuitive worsening of glucose control towards diabetes, which was very concerning to us because many millions of people worldwide consume artificial sweeteners with an aim of satisfying our sweet tooth without having to pay the caloric price, especially people who are prone or already suffer of prediabetes or even type 2 diabetes. And what we could show was that the gut microbiome in some configurations could actually drive a disturbed control of sugar levels in the blood of these mice, which could suggest that in some cases the gut microbiome could actually be bad for you when you consume artificial sweeteners in terms of your sugar management.
That's a landmark study by any standard, so congratulations for doing such important science. And that was back in 2014. So what has research shown since then?
To be honest, this was not only surprising, we were able to prove in that study that consumption of artificial sweetener was not only associated with changes in the microbiome and with a trend towards altered sugar control, but it was actually causative. We did this by transferring microbiomes from mice that were exposed to saccharin or to other artificial sweetener into mice that do not have any microbiome of their own. They are called germ-free mice. And we could see that these recipient mice, which never consumed artificial sweeteners, were developing the same exact disturbances in their sugar control just by the exposure to microbes that were previously exposed to artificial sweeteners. Now, this was very surprising, very important, and also caused a lot of backfire from the industry, as one would expect, but it led to really a rain of follow-up studies by multiple groups, mainly in other mice models and utilizing other formulations of artificial sweeteners, which basically suggested that our observations were correct and that artificial sweeteners were non-inert when given to mice in different contexts and disease states. Now, the biggest limitation of our original study and all of these many follow-up studies was that all of these experiments were conducted in mice, and the billion-dollar question was, can we recapitulate these findings, at least to some extent, in the human setting? And this prompted us to perform a very ambitious human clinical trial, which took multiple years to complete, and was finally published a year ago. And the take-home message from that study, which focused on all 4 commonly consumed artificial sweeteners, was the concept that we've highlighted almost a decade ago was correct when applied to humans.
Those findings, boy, raise alarms on many different levels. So I appreciate you sharing that information with us. Recently, the World Health Organization has expressed concern about the sweeteners and their impact on ability to control body weight and health and even risk for cancer. And one of the counters from people who are in favor of these sweeteners is that the doses provided in these studies are many times what a human might ordinarily consume. What do you say to that?
I would say two important things. First of all, while this concern or may have been true in previous studies a decade ago, in which we all started by testing artificial sweeteners at relatively high doses, with years of follow-up studies and the expanded field, the doses of tested artificial sweeteners were in many cases reduced to those below the recommended daily allowance in humans. And actually, in the controlled clinical trials that completed and published a year ago, doses of 4 artificial sweeteners that we supplemented our participants were well below the recommended daily allowance. And still, in some people, we could see that the seemingly inert compounds were able to reproducibly change the microbiome and to change it to a configuration which could induce alterations in blood sugar control in these individuals, and upon transfer to mice that have never seen artificial sweeteners, these microbiome configurations could also induce identical disturbances in blood sugar control in transferred mice. So I think that our studies and a plethora of other studies generate knowledge suggesting that artificial sweeteners may not be inert to the human body, even when consumed in conservative doses.
Well, thanks for that clarification. You mentioned some pushback from industry. Tell me what form that took, and was there a particular player in industry that was doing the pushback, and did you think that their concerns were valid?
Well, I think that it's a natural part of an evolution of a set of discoveries that have a direct impact on human health and human nutrition, and also on, of course, economic considerations. Some of the claims were less than fair, others were more relevant, and one needs to understand, and I'm saying this both as a physician and as a scientist, especially given our early studies, that you can do the best effort that you can and comprehensively try to tackle a question, but there are always things people can claim that were not done or should be done better. And science is not a one-shot profession. We come up with a discovery and then we build upon it, and others also come in, validate it, build upon it. And this was the process. So I take everything with love. Many other people have experienced a healthy discussion in the industry, and I think we've all matured into understanding, at least in our group, we are not against artificial sweeteners or any other food compound. We are just keen and interested in learning how different food compounds, including artificial sweeteners, impact our body in unforeseen ways. And we focus on the gut microbiome as this huge and poorly explored ecosystem, which we now understand is an integral part of our body and has amazing biochemical capacities to degrade and respond to almost any substance. And with artificial sweeteners, I think that this revelation is actually very helpful to the industry, because with this knowledge and knowledge that is added by others, we are hopeful that by understanding that artificial sweeteners are not inert to the human bodies, we would be able, together with the industry or with whoever is interested in helping this effort, understand which formulations are safer than others, which doses are safer than others, and which are the human populations at risk, which should think twice before they consume these compounds, while others could consume them and enjoy them with relatively little risk. So I think this maturation of our knowledge and maturation of the field is in general a very positive one.
So with respect to the impact of these sweeteners on the microbiome, is there reason to have special concern about children?
When we conduct human clinical trials, we're always hypersensitive about special populations at risk, such as children, people who are incarcerated, and so on and so forth. And we usually tend, at least in groundbreaking first-in-human trials, such as the one that we've conducted with artificial sweeteners, we try to avoid these at-risk populations just so our initial observations are based on populations that are not at special risk. So children, are always a special population which we're even more concerned about than a general population. Our own study was not inclusive of children, so our inclusion criteria, including healthy adults 18 years of age and older. So with respect to children, I can say that our own data did not explore them to date, but in general, we are always especially concerned about children. Any potential metabolic perturbation during this critical window from children is of special medical concern to us.
Let me ask you one final big picture question. So the defenders of these products are basically implying, if not sort of stating directly, that the pursuit of sweetness is a reasonable thing to have, and that these products can deliver the sweetness that people enjoy, but potentially without negative effects. That's the position. But what about that basic proposition that the pursuit of sweetness is a reasonable thing to do? Because couldn't one say that this pursuit, whether it's delivered, whether the remedy to wanting to have more sweetness comes from either sugar or artificial sweeteners, there could potentially be harm? Wouldn't it make sense to just gradually reduce the level of sweetness in the food supply so that people become accustomed to less sweet taste overall?
I couldn't agree more to your statement. I have to say that our findings related to the non-inertness of artificial sweeteners and the potential adverse impacts on human metabolism do not imply in any form or shape that we recommend that people convert back from artificial sweetener into increased consumption of sugar. We know from endless medical literature that sugar is probably an ultimate evil when consumed in excess and is directly responsible for contribution to multiple human diseases, including obesity, type 2 diabetes, fatty liver, and even other diseases, including cancer. So going back to sugar would be a big mistake. But I totally agree with you that positioning the public opinion as one which has to choose between two sweet solutions, either sugar or non-nutritive sweeteners as the only options, basically is biased because we as a post-industrial revolution human population have dramatically increased our sugar consumption. And I don't think that we need to choose between these two options. I personally opt to go back to water rather than choose between these two options, which carry along probably adverse health consequences, at least in some human populations.
You and your colleagues are doing path-breaking work, and it's wonderful for you to join us today and to share this with us. So thanks so much for being with us today.
Pleasure chatting with you.
So our guest today has been Dr. Eren Elinav from the Weizmann Institute of Science in Israel. We have recorded a series of podcasts on sweeteners that can be accessed by putting the leading voices in food into your browser. Thank you for listening, and thanks to Deborah Hill for her great work as producer of this series. If you'd like to subscribe to the Leading Voices in Food podcast series, you can find it on Apple Podcasts, Google Podcasts, or your favorite podcast app. Podcasts and transcripts are also available on the website of the Duke World Food Policy Center. This is Kelly Brownell.